Welcome to the Deep Dive. Today, we're starting, a really in-depth exploration of something truly monumental in the history of science.
Yeah, something that didn't just tweak human health, it fundamentally changed the whole game.
Exactly. Our mission here is to unpack the incredible life-saving story of antibiotics. This is genuinely an amazing story.
It really is. And look, it's more than just another medical topic. This is, you could argue, the foundational pillar of all of modern medicine.
Right.
And I think to really get the scale of it, we need to start with a pretty mind-blowing statistic. It sort of sets the stage for this whole deep dive.
Okay, let's hear it.
Yeah.
Because the historical impact we're talking about here is just massive.
It is. So antibiotics contributed so significantly to improving human health and critically increased longevity that they're often credited with nearly, get this, doubled the human lifespan over the 20th century.
Doubled. That's staggering. It really underscores the profound impact we're discussing. A genuine scientific wonder.
Absolutely. And it's a transformation that's so complete, it's actually hard for us now to really imagine the sheer terror of the world before antibiotics.
Yeah, tell us about that. What was it like?
Well, before this huge scientific advancement, everyday things were potentially legal. Childbirth. Hugely risky because of infections like puerperal fever.
Right.
A simple cut, maybe getting nicked while cooking, or even a toothache turning into an abscess. These things were often fatal. Why? Because of the bacterial infections that inevitably followed.
I was reading about surgery back then. It sounds like the procedure itself was only half the battle.
That's a good way to put it. The real gamble was whether the patient would survive the almost certain infection afterwards. Infectious diseases we barely worry about today, like pneumonia or sepsis, were basically untreatable death sentences for many.
So these drugs weren't just an improvement, they were a revolution. Literally life or death for millions, potentially billions.
Precisely. We went from being, pretty much defenseless against these microscopic threats to having a targeted weapon. And it's a story that has roots going way back, thousands of years of observation, really, but it culminates in this famous accidental scientific discovery.
Okay, let's get into that history. So, long before sophisticated labs, people were already sort of stumbling onto things that worked against infection.
Oh, absolutely. This idea of using natural stuff to fight infections is ancient. We've got evidence, like the Ebers Papyrus from ancient Egypt, maybe around 1550 BC.
What does that say?
It describes using things like moldy bread and certain medicinal soils on wounds. They clearly saw something was helping, even if they had no clue why.
So it was trial and error. Like, hey, this moldy bread seemed to work last time.
Pretty much empirical observation without any understanding of the microbiology involved. It's fascinating. And we even have actual archaeological evidence.
Oh yeah? Like what?
Researchers found traces of tetracycline, which is a well-known antibiotic class we still use in human bones from ancient Nubia.
Wow, old.
Dating back to somewhere between 350 and 550 CE.
So hundreds of years ago, how did it get there? Were they taking pills?
No, not pills. The theory is they were consuming it regularly, maybe unintentionally. It might have been in their grain storage or possibly even in their beer.
Beer? Seriously?
Yeah, the brewing process might have involved grains contaminated with Streptomyces bacteria, that's the natural source of tetracyclines. So, accidental antibiotics.
That's incredible. But, okay, that's ancient history. The real shift, the move towards actual purified medicine, that started much later, right? Early 20th century.
Correct. That required a huge leap in chemistry and a different way of thinking. We needed to move beyond just applying moldy bread.
And that brings us to Paul Ehrlich, doesn't it? He sort of laid the conceptual groundwork.
He really did. Around 1910, Ehrlich gave us this idea of the magic bullet, that's his term. He was searching for a chemical compound that was selectively toxic. Meaning it would kill the pathogen, the bad bug, but leave the host, the human cells, unharmed. That was the dream.
And he was working with dyes. That seems like an odd starting point.
It does, but it was clever. He noticed some dyes would stain bacterial cells, but not human cells. So he thought, okay, maybe we can find chemicals that target bacteria specifically, just like these dyes do.
And did he find one?
He did. His big breakthrough was salvarsan. It was a synthetic drug, actually arsenic-based, which sounds scary.
Yeah, arsenic doesn't sound very selectively toxic.
Well, it was toxic, no doubt. But it did work against syphilis, which is a huge problem. So it was a proof of concept, even if it wasn't perfect. It showed you could target disease with chemicals.
Okay, so Ehrlich sets the stage. Then who comes next?
A couple of decades later, we get Gerhard Domack in the 1930s. He developed Prontosil.
And what was special about Prontosil?
It was the first commercially available antibacterial drug, a sulfonamide or sulfa drug. And crucially, it was much safer than salvarsan. It really cemented the idea that synthetic chemistry could produce these vital medicines. It was a major scientific advancement.
Before we jump to the really famous discovery, you mentioned something called pyosinase earlier. What was that about? It sounds like an early attempt that didn't quite pan out.
Right. Pyosinase is an interesting side story. It was actually an extract made from a bacterium, Pseudomonas aeruginosa. And believe it or not, it was used in hospitals back in the 1890s.
So even before Ehrlich, they were trying to use bacteria against bacteria.
They were. It was probably the first clinical attempt to harness this natural microbial warfare. But the problem was it was a crude extract. Meaning it wasn't one pure substance. It was a complex mix of molecules from the bacteria. So its effects were really inconsistent. Sometimes it worked a bit, sometimes it didn't, and it often caused pretty bad side effects. It wasn't stable, hard to standardize. You get the picture.
Yeah, sounds unreliable.
Very. So once the purified, more predictable synthetic drugs like Prontosil came along, pyosinase just faded away. It really highlights why moving to purified single compounds was such a critical step for modern medicine.
Okay, that makes sense. And that perfectly sets up the moment everyone knows, or thinks they know. Alexander Fleming in 1928 and penicillin.
Ah, yes, the legendary story. Alexander Fleming, a Scottish bacteriologist, he comes back from holiday.
Finds a messy lab bench.
Pretty much. He noticed one of his petri dishes, growing Staphylococcus bacteria, had become contaminated with a mold. A common blue-green mold, which was later identified as Penicillium notatum.
And instead of just tossing it out, like most of us probably would,
he looked closer. He saw this clear zone, like a halo, around the mold colony. The bacteria just weren't growing near it.
the mold was killing them off.
Or at least stopping them from growing. It was secreting something. That observation, that was the crucial moment of scientific discovery. Not necessarily finding a ready-to-use drug, but seeing this natural antagonism in action right there in the dish.
Its hail is this massive medical breakthrough, this revolutionary invention. But there's a key part of the story often glossed over, isn't there? Fleming couldn't actually turn it into medicine right away.
That's absolutely critical. He discovered the phenomenon, he named the substance penicillin, but he really struggled to isolate and purify it.
Why was it so difficult?
Well, the penicillin molecule itself is quite unstable, especially in its raw liquid form. It breaks down easily, particularly in acidic conditions. Fleming just couldn't get a stable, potent powder out of his mold juice, basically.
So for about 10 years, it was more of a lab curiosity.
Exactly. He used it in the lab, sometimes to help isolate bacteria that weren't affected by it, but as a mass-produced drug, no. That required a whole different team and a different set of skills. The real medical breakthrough in terms of a usable medicine came later.
Right, and that's where Howard Florey and Ernst Chain come in, the Oxford team. This is the second act, turning penicillin from that lab curiosity into a world-changing drug.
Yes, this was in the early 1940s. World War II is raging, and there's this desperate need for something to treat battlefield infections, which were killing huge numbers of soldiers.
So the war provided a massive push.
It's immense pressure. Florey, Chain, and their team picked up Fleming's research. Their genius was really in the chemical engineering, the process development. They figured out techniques like freeze-drying, working in non-acidic conditions, ways to finally purify and stabilize penicillin.
And they tested it on a person.
They did. In 1941, they gave the first human dose to a policeman in Oxford who had this terrible widespread sepsis after a simple scratch from a rose thorn.
How'd it go?
Initially, spectacularly well. He started improving dramatically, but their purified supply was tiny. They just couldn't make enough, fast enough. Tragically, they ran out and the infection returned and he died.
Oh, that's heartbreaking. But it proved the concept.
It did. It showed that purified penicillin worked in the body against severe bacterial infections. The potential historical impact was suddenly crystal clear. It just needed scale.
And that's where the government stepped in, US and UK.
Yes. The potential to save soldiers was too huge to ignore. There was this massive coordinated effort, particularly in the US, involving the government, universities, and pharmaceutical industry giants to figure out mass production.
Using things like deep tank fermentation, right?
Yeah.
Finding better strains of the mold.
All of that. They screened different penicillium strains, eventually finding one on a moldy cantaloupe in Peoria, Illinois, believe it or not, that produced much more penicillin. They optimized the whole process.
And by 1943,
By 43, enough penicillin was being produced for the Allied military. It had a dramatic effect on survival rates from wounds, sepsis, amputations. And then by 1945, as the war ended, it started becoming available to the general public. That's when it truly became this medical wonder.
and the success of penicillin, this thing that basically came from mold, it just opened the floodgates, didn't it? It kicked off what we now call the Golden Age of Antibiotics.
Absolutely. The Golden Age of Antibiotics, roughly the 1940s through maybe the early 1970s, was this incredibly exciting, productive period for drug development.
What drove it? Just the success of penicillin.
That was the catalyst. Suddenly, everyone realized nature was full of these microbial weapons. So scientists started screening everything, especially soil samples.
Why soil?
Soil is just teeming with microbes, bacteria, fungi, all competing fiercely with each other for resources. So the thinking was they must be producing chemicals to fight each other off. Natural antibiotics.
Like a hidden chemical arms race under our feet.
Exactly. And this screening effort was incredibly fruitful. It was led by figures like Selman Waxman, who actually coined the term antibiotic in its modern sense.
Right, you mentioned him defining it as something produced by a microbe that inhibits or kills other microbes.
Precisely. And Waxman himself, screening soil bacteria, discovered streptomycin.
And streptomycin was a big deal because
It was effective against tuberculosis. TB was a major killer, notoriously hard to treat. Streptomycin was a game changer for that specific infectious disease.
and this whole soil screening approach yielded most of the antibiotics we still use.
Pretty much all the major classes came out of this era. Tetracyclines, macrolides like erythromycin, cephalosporins, all discovered by screening soil microbes, mostly bacteria from the Streptomyces genus. It was an astonishing period of scientific discovery.
Okay, so we have all these different antibiotics. Let's dig into the mechanism a bit. How does this scientific wonder actually work? The microbiology must be clever to kill bacteria, but not us.
It all comes back to that principle Ehrlich dreamed of, selective toxicity. Antibiotics exploit differences between bacterial cells and human cells.
What kind of differences?
Key structural things. Let's take penicillin again as the classic example. We mentioned it targets the cell wall.
Right, bacteria have a cell wall, but human cells don't.
Exactly. Our cells have a flexible membrane, but bacteria, many of them, have this rigid outer wall made of something called peptidoglycan. It's like scaffolding, gives them shape and protection.
So penicillin messes with that wall.
It does. Specifically, it interferes with the final step in building that peptidoglycan scaffolding. Bacteria have special enzymes proteins that cross-link the pieces of the wall together. These enzymes are called transpeptidases, or sometimes penicillin-binding proteins, PBPs.
Ah, okay, so the name gives it away.
It does. The penicillin molecule chemically mimics the shape of the bits the enzyme normally links together, so the enzyme tries to grab the penicillin instead.
Like jamming the wrong key in a lock.
A great analogy.
Yeah.
And when the enzyme binds to penicillin, it gets stuck. It's irreversibly inhibited. It can't do its job of building the wall anymore.
And without a strong wall.
The bacterial cell is fragile. The pressure inside the cell is usually higher than outside, so without that rigid wall to contain it, the cell basically swells up and bursts. Pop.
Wow, so it's directly lethal. Bactericidal.
Yes, penicillin and related drugs, the beta-lactams, are typically bactericidal. They actively kill the bacteria.
That's fascinatingly precise. And other types of antibiotics use different targets.
Oh yeah, it's like having different tools for different jobs. Some, like tetracyclines or aminoglycosides, target the bacteria's ribosomes.
Ribosomes make proteins, right?
Correct. And bacterial ribosomes are slightly different in structure from our human ribosomes. So these drugs can shut down protein production in bacteria without messing up ours too much. Selective toxicity again.
Clever. Any other main mechanisms?
Another big one is interfering with DNA replication. Drugs like the fluoroquinolones get in the way of the enzymes bacteria use to copy their DNA, so they just can't multiply.
It really is a scientific wonder the way these molecules work. And just to hammer this home, because it's so important for public health, this targeting of bacterial machinery means they do absolutely nothing against viruses, right?
Absolutely zero. It cannot be said enough times. Viruses are completely different beasts. They don't have cell walls. They don't have their own ribosomes. They basically hijack our cell machinery to replicate.
So taking antibiotics for a cold or the flu
Is completely useless for treating the virus. All it does is potentially cause side effects for you, and worse, it exposes all the bystander bacteria in your body, many of which are harmless or even helpful to the drug.
which helps drive antibiotic resistance. We'll definitely get to that. But first, let's just reflect on the massive societal shift. What did this medical breakthrough actually do for the world?
The change was almost immediate and unbelievable. Think about hospital death rates from things like sepsis, blood poisoning. They plummeted. Infections after childbirth, like puerperal fever, went from being a terrifying killer of new mothers to a treatable condition.
That alone must have had a huge demographic impact.
Massive. It changed family structures, population growth, everything. And then there are the ripple effects throughout modern medicine.
What do you mean?
Think about almost any advanced medical procedure we take for granted today. Complex heart surgery, hip replacements, organ transplants, cancer chemotherapy, which wipes out the immune system, caring for premature babies.
Okay, yeah. All of those carry a huge risk of infection.
Exactly. None of them would be feasible, certainly not safely, without effective antibiotics to prevent or treat the inevitable bacterial infections that can occur. Antibiotics aren't just a type of medicine, they're the safety net that makes almost all of modern medicine possible.
It's hard to overstate that profound impact.
Yeah.
But there's always a bet in biology, isn't there? The very success of this scientific wonder, using it so widely, created a new problem.
The inevitable consequence. Evolution. Bacteria are masters of adaptation. They reproduce incredibly quickly. Some can double their population in 20 minutes. And they have ways to swap genes.
So every time we use an antibiotic,
We're essentially running a massive natural selection experiment. Most bacteria are killed, sure. But if just a few happen to have some random mutation or a gene that gives them a slight defense against the drug,
they survive.
they survive. And with all their competition wiped out, they thrive. They multiply, passing on that resistance trait. Over time, the resistant bacteria become dominant. That accelerated evolution is what we call antibiotic resistance.
And when our standard drugs stop working against these evolved bacteria, we get superbugs.
Exactly. Superbugs is the common term for bacteria that have become resistant to multiple antibiotics. They represent a huge and growing global threat.
How serious is it right now? Do we have numbers?
We do, and they're sobering. The source data mentioned, just in the US, we're looking at something like 2 million infections and maybe 23,000 deaths every single year directly caused by antibiotic-resistant bacteria.
23,000 deaths a year in the US alone. That's a serious health crisis already happening.
It is, and it threatens to undo so much of the progress we made, all that increased longevity gained in the 20th century. We genuinely risk slipping back towards a pre-penicillin reality for some infections.
Are there specific superbugs people might have heard of?
Oh, sure. MRSA is probably the most famous, methicillin-resistant Staphylococcus aureus.
Yeah.
Then there's VRE, vancomycin-resistant Enterococcus, and some really scary ones like CRE, carbapenem-resistant Enterobacteriaceae. Carbapenems are often our last resort antibiotics for serious infections.
So when bacteria become resistant even to those,
We might have literally no options left. These infections, often picked up in hospitals, can become untreatable, forcing doctors to use older, sometimes highly toxic drugs that we'd abandon for good reason.
let's connect this back to the mechanism we discussed. How does a bacterium actually become resistant to something like penicillin? What's it doing differently?
Okay, great question. One of the most common ways bacteria resist penicillin and its relatives is by producing enzymes called beta-lactamases.
Beta-lactamase, so it breaks down the beta-lactam drugs.
Precisely. Remember we talked about that crucial beta-lactam ring in the penicillin molecule, the part that jams the bacterial enzyme?
Yeah.
Well, beta-lactamase enzymes are like molecular scissors. They specifically find that beta-lactam ring and cut it open. Snap.
And once the ring is broken,
The drug is useless. It's chemically inactivated. It can't bind to the penicillin-binding proteins anymore, so the bacteria can just keep building their cell walls quite happily. This one mechanism, producing these enzymes, is a major way bacteria fight back against our most important class of antibiotics.
that's remarkable. And fascinatingly, you mentioned earlier, this isn't entirely new, is it? Bacteria didn't just invent these defenses when we started using penicillin.
That's a really key point from microbiology. The genes that confer resistance, like the genes for beta-lactamases, they're ancient. They've been around in the microbial world for potentially millions, maybe even billions of years.
How on earth do we know that?
Scientists have done some amazing work. They've looked at DNA extracted from really old environmental samples, like 30,000-year-old permafrost cores from the Yukon.
Wow, ancient frozen bacteria.
Exactly. And they found genes in those ancient bacteria that are clearly related to modern antibiotic resistance genes. They've also studied bacteria from environments totally isolated from humans and modern medicine, like deep caves.
Luchugia cave, for example.
That's a famous one. Bacteria there, isolated for maybe 4 million years, already had resistance mechanisms against many of our modern antibiotics, even drugs they'd presumably never encountered.
So what does that tell us?
It tells us that microbes have been waging chemical warfare against each other for eons. Developing toxins and defenses, antibiotics and resistance is just part of their natural evolutionary game. The resistance genes were already out there in the environment.
What changed? Why is it a crisis now?
What changed is us. We started mass producing these natural compounds or synthetic versions and using them on an unprecedented scale. In medicine, yes, but also hugely in agriculture.
Right, you hear about antibiotics in farming.
It's a massive factor, giving low doses of antibiotics to livestock to promote slightly faster growth or prevent infections in crowded conditions. That creates this enormous selective pressure of breeding resistant bacteria on farms.
And those resistant bacteria can then spread.
They can spread through the environment, through the food chain, and the resistance genes themselves can even jump between different types of bacteria. We basically took a slow, natural process and put it into hyperdrive through massive overuse and misuse. We created the perfect storm for superbugs.
Which leads us into the really thorny ethical and access issues around this medical wonder. Because the problem isn't just biological, is it?
Not at all. It's deeply tied to global inequality. You have these stark contrasts. In many low-income countries, people are still dying because they can't get access to basic, essential antibiotics for simple bacterial infections.
A tragedy of lack of access.
Exactly. But then in other parts of the world, sometimes even in the same regions or in middle-income countries, you have the opposite problem. Antibiotics are available over the counter, without prescription, leading to rampant misuse.
People taking them for viruses, not finishing the course.
All the things that accelerate resistance, it's like treating these precious medicines like vitamins. And that misuse anywhere fuels the global threat everywhere because resistant bacteria don't respect borders.
So we have lack of access in some places, massive overuse in others, including agriculture. It's a complex global mess.
It really is. And it requires a coordinated response from governments, the pharmaceutical industry, doctors, vets, farmers, and the public. It's a shared responsibility.
Okay, so we're facing this growing health crisis of resistance. And we know the golden age of antibiotics, that period of easy discovery from soil, largely ended back in the 70s. The pipeline for new drugs seems to have dried up. What does that mean for the future of medicine?
It means we're in a precarious position. Finding and developing new antibiotics, especially ones with novel mechanisms that resistant bacteria haven't seen before, is incredibly difficult and expensive.
Why is it so hard now? Have we picked all the low-hanging fruit from the soil?
That's part of it. The easy-to-find compounds produced by common soil microbes have largely been discovered. We keep finding the same things over and over. But there's also a big economic problem.
The market failure issue.
Exactly. Developing a new drug costs a fortune, hundreds of millions, maybe billions of dollars. But for an antibiotic, the return on investment for the pharmaceutical industry is often really poor.
Why is that? You'd think a new drug for superbugs would be valuable.
It is valuable to society, but not necessarily profitable. Think about it. If a company develops a fantastic new antibiotic that works against resistant bugs, what's the first thing public health officials will say?
Use it sparingly. Save it for the worst cases to preserve its effectiveness.
Right. So unlike a drug for a chronic condition that someone takes every day for years, a new antibiotic ideally should have low sales volume. That makes it commercially unattractive compared to, say, a new cancer drug or a cholesterol medication.
That's a serious disincentive for drug development. So how do we overcome that? What scientific advancement pathways are researchers actually pursuing now? Where are they looking, if not just soil?
They're having to get much more creative and go to more extreme places. We're talking deep-sea exploration, sampling sediments from the ocean floor.
Really, there are useful microbes down there.
Oh, yes. The pressure, the cold, the unique chemistry, it fosters unique microbial life. Researchers have found marine organisms like bacteria in the genus Selenospora that produce completely novel types of molecules with antibiotic activity, things structurally different from what we find on land.
So prospecting in extreme environments. What else?
Exploring previously overlooked sources like rare bacteria from intricate cave systems or symbiotic microbes living inside insects or plants. Also tapping into the potential of the microbes already living in and on us, our own microbiome. Maybe some of our beneficial bacteria produce compounds we could harness.
Okay, that's about finding new sources. What about totally different approaches? I've heard about phage therapy making a comeback.
Yes, bacteriophages, or phages for short. These are viruses that specifically infect and kill bacteria.
Using a virus to kill a bacterium. Sounds like fighting fire with fire.
Kind of, but phages are incredibly specific. A particular phage might only infect one strain or species of bacteria. This makes them potentially very precise weapons.
So, less collateral damage to our good bacteria than broad-spectrum antibiotics.
That's the hope. Phage therapy was actually used quite a bit in Eastern Europe, especially before antibiotics became widespread. But it fell out of favor in the West. Now, with resistance rising, there's huge renewed interest.
Can we engineer them?
Absolutely. Using modern genetic tools, researchers are trying to create custom phage cocktails, maybe even genetically engineered phages, designed to target specific, difficult superbugs. It's a really exciting area of scientific advancement.
What about using our knowledge of bacterial genetics? Can we design drugs more rationally now?
Yes, genomics and synthetic biology are playing a huge role. We can sequence bacterial genomes very quickly now. This helps us identify essential genes or pathways in bacteria that could be good drug targets.
And maybe find resistance mechanisms before they become widespread.
Potentially, yes. Genomics helps track resistance. Synthetic biology allows us to either engineer microbes to produce novel compounds or even design and build completely new antibiotic-like molecules from scratch in the lab. Or we can take existing antibiotics and chemically modify them to overcome resistance, creating next-generation versions.
The ultimate goal being more targeted drugs, narrow spectrum.
That's a major aim. Instead of carpet bombing our entire microbiome with broad-spectrum antibiotics, developing drugs that only kill the specific pathogen causing the infection would be a massive leap forward for human health and would hopefully slow down resistance development too. It's all part of securing the future of medicine.
So while all this exciting research is happening, which might take years to yield new drugs, we absolutely have to protect the antibiotics we do have now. That comes down to stewardship, right?
Stewardship is absolutely critical. It's the immediate, essential action we can all take. It starts with doctors and healthcare providers by using diagnostics more effectively. We need rapid tests that can quickly tell a doctor if an infection is bacterial or viral, and if it is bacterial, which specific bacterium it is and what drugs it's susceptible to. Prescribing antibiotics only when necessary and prescribing the right antibiotic is key.
Avoiding that just in case prescription for a cough or cold.
Exactly. And reducing the use of broad-spectrum drugs when a narrower one would do. And then there's the patient's role.
Finishing the full course of medication, even if you feel better.
Yes. It's so important. Stopping treatment early might leave behind the slightly tougher, more resistant bacteria. They survive, multiply, and next time, the infection might be much harder to treat. Completing the course helps wipe out as many of the bacteria as possible.
And on a bigger scale, beyond individual actions.
We need major changes in policy, significantly reducing, ideally eliminating, the use of antibiotics for growth promotion in agriculture is vital. Improving sanitation, hygiene, and access to vaccines globally is also crucial. Preventing infections in the first place reduces the need for antibiotics altogether.
It sounds like it requires massive global cooperation. Tracking resistance, sharing data.
It does. And finding ways to fix that broken market for new drug development. Maybe governments need to offer prizes or guarantee purchase agreements to incentivize the pharmaceutical industry to reinvest in antibiotic R&D. It's a complex puzzle needing international effort.
Looking back then, this whole journey, this life-saving story, It starts with Alexander Fleming's contaminated petri dish, this moment of accidental scientific discovery.
Yeah, a moment that led to the Golden Age of Antibiotics, transformed public health, and gave us modern medicine as we know it.
But now we're in this critical fight against antibiotic resistance and superbugs, trying to protect that legacy.
It truly remains one of the greatest medical breakthroughs, a profound scientific wonder. Antibiotics fundamentally reshaped human civilization, enabling the population growth, the increased longevity, the quality of life we often take for granted. Their profound impact is almost immeasurable.
And as we look towards the future of medicine, trying to develop new solutions and protect the old ones, we run right into that difficult ethical question you raised earlier.
We do. It's the central dilemma, really, in preserving this scientific wonder. How do we as a global community balance two critical needs?
Which are?
On one hand, the undeniable moral imperative to ensure widespread, equitable access to existing life-saving antibiotics in low-income nations where people are still dying from basic infectious diseases. They need these drugs now.
Right.
But on the other hand, the absolute necessity of implementing strict stewardship, controlling use everywhere, reducing agricultural dependence to slow down resistance and protect these precious drugs for the long term, for everyone, for the future of medicine.
Access versus preservation.
Finding that balance, ensuring equity today while safeguarding effectiveness for tomorrow, that's the ultimate challenge. How we navigate that will determine whether we hold on to the incredible gains that nearly doubled human lifespan that antibiotics helped us achieve.